everyone, welcome to my show. My name is Elnaz. I have a PhD in cellular and molecular biology and I'm a bioinformatician. In this video, I'm going to talk about alignment. What's the basic concept and algorithm of alignment in RNA sequencing data analysis, which tools work, how, and etc. Well, when we are going to have our RNA sequencing experiment, we usually split the RNA molecules and DNA molecules to short fragments and then send them for the sequencing. Since the splitting the RNA and DNA molecules is done by the enzymes, so we are sure that we have short molecules. After sequencing, we have short reads which have overlaps together. So this overlap and also the genome, the reference genome, is used for the alignment. And the mission of the alignment is that to determine which short read belongs to which part of the genome and which gene exactly. For like some experiments like the whole transcriptome or transcriptome analysis, we want to know which short read belongs to which gene. So it's used for the quantification. So this is what alignment does. But what's the exact method? Let's say we have we have an enormous genome and we have millions or billions of short reads, and we want to know which read belongs to which part of the genome. So we can do this, but there's a better way which saves us time and also the memory usage, and it's called genome indexing. Genome indexing determines that which part of the genome or the chromosome or the gene has what functionality, let's say. It's something like that. So for indexing, the main strategy is the suffix indexing, and for this the method which is used is suffix array. Suffix array is a computational method, the mathematical method in which string, each character is treated and to know what's the character beside, etc. It also works for the genome alignment and genome assembly, because we have to assemble the short reads to know which reads participate in one gene, or let's say in one part, and special one part of the genome. Suffix array is used for the alignment of the genome using some special determinants. For example, the position of the suffix, the overlap of nucleotides with each other, the overlap size, and it determines the leftmost position of each nucleotide. And this is how we determine the exact nucleotide array of a short read, and then we can see and say what's the coverage and what's the most coverage between one short read and another short read, to evaluate the assembly and make a bigger part from those short reads. But the first time that suffix array was introduced, well, it made many things easy in RNA sequencing data analysis, but it was famous because of the high memory usage and also a long runtime. So another algorithm was introduced, which was called full text index in minute space, or FM index. FM index is based on BWT algorithm. It's similar to suffix array, but let's say it has shorter runtime and also smaller memory usage. And also, since in many of the aligners today, using BWT algorithm, the step for indexing is removed for developing the suffix indexes is removed, and the pre-assumed suffixes are used. So this takes shorter time and makes the alignment easier. As you can see in here, the BWT-FM index for reference genome works like this, like which nucleotide is which position in the query of the genome, and what's the alignment and what's the result of the alignment. And also another example of BWT alignment for a word like banana, and to determine how banana works and what is the position of banana, what's the position of the nucleotide, or let's say the word of the left word and the right word, and this is how we determine the exact sequence of a short read in alignment. You may have heard that alignment has different types. One of them is alignment against a reference genome, another one is alignment against reference transcriptome, and last one is pseudo alignment. In this video, I'm not going to talk about pseudo alignment because it has a special method for doing the alignment, but in the next videos, I will have one video special for pseudo alignment. But in this video, I will discuss about reference genome and reference transcriptome alignment. So we have several alignment tools. When we are going to start an RNA sequencing data analysis, we face this issue. Okay, I have several tools like HiSat2, STAR, TopHat2, ROM, BOTI2, Callisto, Salmon, Selfish, and like this. But which tool I should use? Well, if you ask me, I would say this is a critical question, but it depends on what you are looking for in your study. What's the aim of the RNA sequencing data analysis? Do you want to find the variations? Do you want to find the differential expressed genes, or any other aims do you have? What's the aim of your study? Keep this in mind, and let's say alignment against reference genome and alignment against reference transcriptome work differently and give us somehow different results. In alignment against genome, I assume that I have a gene with 3 exons, and there are introns also in this gene, and I have a lot of short reads. So using FM index or also suffix array, which work, they both are sufficient and efficient method. So I align the short reads to these exons, and I find that in some of tools which use alignment against reference genome are BWA and BOTI2. As you can see from the name of these alignments, they both use BWT algorithm for the alignment. So where they align the short reads for the exons. This is good because we can determine that each short belongs to different parts of the genome, and we can say that okay, there are several exons in this one gene, and even if there is an alternative splicing, we will determine and we will recognize that if we do alignment against reference genome. But also, this method for the alignment has its own disadvantages. For example, it has a long runtime, it has large space, it's really complicated and difficult because we have, as I said, enormous genome, and we want to align all those short reads against this genome. There's a big runtime, there's a big memory usage, and also one of the let's say bad problems coming up with alignment against reference genome is that it may develop pseudogenes because of its ability to align to whole genome. For example, let's say some of short reads belong or aligned to an intron, but the intron wasn't in the gene and it wasn't an alternative splicing. So what would we do? So these are some issues. And people who are looking for, let's say, special genes, somehow they prefer to use alignment against reference transcriptome. Reference transcriptome, we have some tools in this method, like TopHat2, STAR, and HiSat2, and also I have read some articles, and I know, and I have tried also STAR for reference genome, and it works, but I'm not sure about which work better. But I will discuss the same issue in the next video, so you will get the answer for this question in the next video. So how does it work? If we have billions of short reads that we want to align them but against transcriptome, we will use them, and the method for finding the coverage and the overlap between short reads to assemble the whole transcriptome using the short reads. Then we will align those short reads to the assembled transcriptome. So as you can see, the aligned reads exactly shows the reads which are coming from one gene, and there is no information about the introns. But also I should mention that there is no information about the alternative splicing or something similar. So if there is an isoform, a novel isoform, we couldn't realize that using alignment against reference transcriptome. But this is very good because it has a shorter runtime, and also it uses smaller space from the memory of your computer system, and it has its own, let's say, advantages and disadvantages. But if you want to see how many copies you have from one special read or RNA molecule, mRNA molecule, for example, you can use this for quantification because you have transcriptome, you don't need introns, and there you go, you can have the count of your reads. So, as I mentioned before, for deciding about which tool or which method we should use for alignment in the RNA sequencing pipeline for the analysis of our data, it's always good to know what's the aim of our analysis. And also, I would recommend you to go for the tutorials or manuals of each tool and read about them and see what are the exact information they can help us to achieve when we are using those alignment tools. I have used several articles and web pages for preparing this presentation and this video, which I'm bringing them in here, and I also put them under the video. I hope you liked this video and it was helpful for you. You're always welcome to make comments, and please share this video with people who are searching for it. Thank you, have a good day, and I will see you, I hope to see you in the next videos. Bye.